Hi. In my last lecture, I told you about experimental animals, and today we shall be continuing our studies with other animals. We started from E. coli and we reached up to rat. Rat, guinea pig, rabbit, mice, mouse, they are all rodents. And the rodents, they have almost similar habits. So they require a similar type of animal house. They require similar type of the laboratory care. Rat, why do we take rat? I told you that the sufficient background information is available. They are easy to maintain, they are easy to handle, they are humble, they do not bite, they do not attack, and they breed under laboratory conditions. That's most important. That they can breed under laboratory conditions. If we are working on some genetical problem, we can get the F1, F2, F3 generations. If we are working on some another issue related to genetics, we need different generations, we can get those models. But how we are going to procure them? We can procure them from registered animal suppliers. I told you CDRI, All India Institute of Medical Sciences, IVRI, and other sources, they provide you the standard healthy animals, post-weaning animals. That is, the the uh, the animals that we use should mainly be of a reproductive age. They should be adults. They should not be juveniles. They should not be old enough. They should not be overweight. If it is so, we are going to get wrong results. So they should be of a proper weight. They should be of a proper age, and they should be maintained under laboratory conditions properly. How we can maintain them? Almost all research institutes have got separate animal houses, and each animal house has got different portions or different compartments. One compartment is to maintain records, another compartment is to change your clothes, the third compartment is to keep the animals, and the fourth one is a store where you keep water bottles, extra animal cages, extra food, and the other accessories which are required. The most important thing for an animal house is that it should run on a constant temperature, on a constant humidity. If the temperature is too low, you will not get good results. If the temperature is too high, you will not get good results. The best way is to maintain these animals between 22 to 25 or 27 degrees centigrade, and the standard relative humidity should not exceed 60%. And the light available to them should be 12 hours light and 12 hours dark. That is the standard protocol that we maintained for rat or guinea pig or hamster. So it's not very difficult. You can have a animal house, you can have an animal room, and the animal can be maintained under these conditions. Now, next thing after procurement is their quarantine. When you procure the animals from a commercial supplier, you should allow a quarantine of at least 15 days, 2 weeks. You should not start the experiment just as you have picked the animal from the supplier and you start experiment. No. First acclimatize them to the animal house conditions, to the new conditions that an animal is facing. The conditions at IVRI or CDRI or AIMS were different than your laboratory conditions. So allow these animals to quarantine and allow these animals to acclimatize with those conditions. When acclimatization has occurred, then you can proceed with the experiment. During the experiment, pre-experiment, post-experiment, how you are going to maintain the animal. The animal is to be kept in a cage. Different types of cages are available. There can be a stainless steel cage, there can be a propylene cage, There can be a propylene cage for a small mice, there can be a propylene cage for a big mice. So these cages differ. These cages differ in size. Generally, we maintain a single animal in a single cage. So, a small propylene cage with a water bottle, automatic water bottle, is provided. These cages have got two compartments. One compartment is meant for water, another compartment is meant for the food pellets. So, you provide them a bed also. The bed should be of a nature that absorbs their urine and feces. The cage should not be dirty. The cage should be cleaned almost every day. The water bottle should be cleaned every day. Right? The food should be replaced every day. These are some of the standard conditions. Remember, if you will clean and if you will maintain the animal in a hygienic condition, you are supposed to get very good results. But if you will not maintain your animals in hygienic conditions, I am very much sure with my experience of 50 years that you are going to get very bad results. For an experimental scientist, animal model or the rat or whatever you have selected is the most important thing in your life. Go to the animal house every day, see whether animals are having healthy conditions, there is no comorbid conditions, there is no diarrhea, there is no bleeding, there is no external symptoms of any type, and you can find out, oh yes, good, your rat is healthy. Then you should change your clothes, have the animal house clothes, change your shoes, put a mask on your nose, put a cap on your head, and clean the animals. So you can maintain them under cages. Those cages may be of stainless steel, the cages may be of propylene, or even if the cages may be of wooden cages, may be there according to the specialized conditions that you need. But the propylene cages are easy to clean, steel cages are easy to clean, so we use them. There are other types of cages, we call them as metabolic cages. They are meant for collecting the urine sample or feces of the animals. The metabolic cages separate the feces and the urine. You collect the urine in a test tube or in a glass bottle. You collect the feces in a petri plate or petri dish, and you can use them. So such type of specialized cages are also available. We call them as metabolic cages. There are separate cages for rabbits. There are separate cages for dogs. There are separate cages for monkeys. There are separate cages for rats or small animals like guinea pigs and all that. So you procured them, you quarantined them, and now you have started maintaining them under standard laboratory conditions, and now you proceed further. What type of you can supply the water supply which is available in your laboratory, that's good enough. Then otherwise you are going to put something in the drinking water like alcohol or like any solvent or like any pollutant or any chemical, that is something. Animal houses should be hazard free. Take it granted that there are sufficient arrangements for fire control, there are sufficient places or separate places to keep the hazardous chemicals. So a trained veterinarian, I told you yesterday also, trained veterinarian should take care of these animals. So after quarantine, after maintaining them under standard laboratory conditions, you can proceed with your experiment. Then the type of the food. Food is very important for animals. What type of food you are going to provide them? It's not like this you provide them bread, no. They will be malnourished. You provide them rice, cooked rice, no. You provide them grams, no. Nothing doing. You should procure the standard laboratory feed which you know that it contains this much amount of protein, fat, sugar, carbohydrate, minerals and ash and all that. You should know it. So there are specific requirements for rat, there are specific requirements for guinea pig, specific requirements for rabbit. So you get rabbit food, you get rat food, you get mice food, you get hamster food, you get guinea pig food separately from animal food suppliers. So you should be very particular about it, that you are feeding the animals with the proper type of food. The bed you provide them, it should contain of a rice husk. You can use the paper cuttings, you can use the small pieces of thermocol which can be removed and thrown. So it is to be like this, that it is going to be disposed of. So this is about food, this is about water, this is about that maintenance. Do not handle them in a very rough way. You should be very humble to your animals. You should love your animals. You should be very humane to your animals, right? So the person who goes to animal house every day, inspect the animal house every day, he knows how to the animals are to be maintained. He or she knows it very well. Then after that, the personal hygiene is very important. Personal hygiene means your hygiene. You should take care of. You should wear a laboratory coat. You should cover your head. You should cover your nose. You should wear the gloves. This all comes under personal hygiene. You should observe the personal hygiene. Then the animal husbandry conditions are like caging, food, weather, sanitation. You should have separate buckets for the waste disposal, for the disposal of the husk, for the disposal of the feces, and that all comes under animal husbandry conditions. Then there are certain SOPs, standard operating procedures. When you perform an experiment, you should follow certain standard operating procedures, right? If you are making some pharmacological or toxicological observations, you should know that what is the proper dose to be given, how it is to be given, whether you are going to administer the dose intraperitoneally or intramuscularly, or you are going to give the dose through mouth. We call it as garbage or force feeding, or you are going to give it in the drinking water, right? Say for example, arsenic you want to study, and the arsenic can be mixed with drinking water, but the problem is you should record the amount of the water taken by each rat every day. Then only you can find out that how much of arsenic has been consumed by the drinking water by the animal. So the administration of the experimental drug is very important. It's very important how you administer it, because the effect of an intraperitoneal injection, intramuscular injection, or the effect given through mouth will be different. And then these effects will be determined by the type of the food you have given. If the food is protein deficient, the effect will be different. If the food is lipid deficient, the effect will be different. The effects will be different in a male animal and the female animal. The effects will be different in a young animal and the aged animal. So you have to be very cautious about it, because these experiments, the results which you are going to get, they will be used they can be extrapolated to man for pharmacological studies, for the drug testing and other studies. So this comes under SOPs. And if you are going to, you may have a plan to study the animal after single dose of a drug, after 15 days of the administration, after 30 days, 60 days, 90 days, 6 months, like that. It depends upon your standard, upon your objective, what type of objective you have. So after completion of the experiment, the another problem starts. After completion of your experiment, how you are going to deal with the animal. You will anesthetize it, or you will euthanize it. Many times we anesthetize the animal with sodium pentobarbital, which is now banned in India. It is not easily available. Or we use solvent ether to anesthetize, or we use a mild dose of chloroform, or such type of, the anesthetizing agents are available. Some new agents are also available which do not affect the other metabolic activities of the animal. Sometimes you are to study the mechanisms like oxidative phosphorylation, single transduction, and others. In that case, the anesthesia is not allowed. You decapitate the animal. Decapitation means a removal of the head. You cut the animal from throat and you collect the tissues to perform the experiments, or you collect the blood just by squeezing the animal. Though it is it's not a very desirable procedure, but sometimes you are to do it. So you should know what are the anesthetic and euthanatic chemicals available, and how you can treat the animal with these type of anesthetics or the euthanatics. Then we come down to the other type of studies. I told you should be very humane to the animal. Other type of animals which are available are transgenic animals. Nowadays we get the knockout animals. Knockout animals means we know that this particular drug is to be metabolized by cytochrome P450, or the specialized isoform of this enzyme. So we can genetically manipulate an animal which are deficient in cytochrome P450. We can manipulate an animal which is deficient in any enzyme or any gene, to be more specific, any gene. So such type of animal, though they are very expensive, but such type of animals are available commercially, or you can produce yourself under laboratory conditions yourself, but you should be expert enough to do that. You should have a very good knowledge of genetics and all that. Then you can do that. This is called as the transgenic animals, or in other words, they are called as knockout animals. The gene knockout. You are interested in methylothionein, and you want to know what is the function of methylothionein. For a control, you need a methylothionein knockout mouse or knockout rat, means where methylothionein is absent. So such type of animals, which we call them as transgenic animals, they are also available. There is one very important phenomenon that we call as the transdifferentiation. Transdifferentiation means that sometimes you need, say for example, a particular tissue from a particular person or a group of persons, and you just take the biopsy sample, and that biopsy sample is transdifferentiated into a suitable animal. Generally it happens with the golden hamsters. Golden hamsters, say for example, you want to study the liver cancer, and you just take a liver cancer tissue, you can implant that liver cancer tissue into the golden hamster. You can implant it inside, you can put it on the muscles of the golden hamsters, and you can stitch it up, and then it will be growing in the golden hamster. Those cells will not die, they will be growing in the golden hamster. Whenever you need them, they will be not only maintaining, but the growth will be there. You can, whenever you need them, you can take them out and use them. Such type of animals are also available. So the animal studies or the animal manipulations are at the height of biology or biomedicine. So this process is called as transdifferentiation. You should differentiate between transgenic and transdifferentiations. Then there are the last thing that is very important is, because the rat has consumed some hazardous chemical, so the rat is to be properly disposed of. It is a biohazard, and in many countries there are very strict laws to maintain the biohazards. You cannot throw it outside the laboratory. It's no way. You should put that in a specially labeled bucket, which have got the sign of a biohazard, and it should be given to the biohazard vehicle. In many countries, the vehicles, they visit the laboratories and they collect these dead animals, and they take away and they micro incinerate it. They burn them, right? So say for example, if that rat is eaten up by a dog, then there are chances that the dog will be suffering from the effects of that chemical. And if that dog bites a man, then the things becomes very dangerous. So the disposal of the experiment animals is most important thing. And since I told you that it is a biohazard, are laws governing the biohazards waste disposal. So you are to give it to a suitable agency who is dealing in the management of the biohazard waste. The agency will micro incinerate it and then it. The next important thing is to maintain the record right from the day one when you got the animal into your animal house until the death of the animal, till its sacrifice by you for some good purpose. Every piece of record is to be maintained, right? In India also we observe good laboratory practice. We in other countries also they observe good laboratory practice, and under good laboratory practice comes the maintenance of the record. What was the weight of the animal, whether there was any comorbid condition in the animal, whether there was any other problem, there was any tumor in the animal apparently visible. Sometimes the tumors they develop in the animal and you find that the tumors are, in that case the animal has to be discarded. If there are animal exhibits the diarrheic conditions, animal is showing diarrhea, then the animal is to be discarded. If the animal is having secondary infection of a tapeworm or of ascaris or any other bacterial or parasitic infection, the rat is to be discarded. So these are the rules which are meant for rat, mice, mouse, hamster, guinea pig, rabbit, all these rodents. For monkeys, of course, you need a separate monkey house, a big monkey house, and you need the permission from the Department of Forest and Wildlife. You need permission from other suitable ministry, and all those birds which are endangered or all those animals which are in the red data book of IUCN, you are not supposed to work upon them. You cannot keep them under captivity. You cannot work them unless and until you have got a specific permission. So only the pharmaceutical companies, very reputed pharmaceutical companies, or the highly sophisticated research institutions, they are allowed to keep the monkeys. And again, monkeys, what type of monkey, whether it is rhesus monkey or whether it is Hanuman monkey or whether it is chimpanzee or all like this, the people work on them and they help us in understanding many of the important mechanisms. But it is it is not advisable to work upon these dangerous animals. You need a permission for that. So it's a very difficult task. I told you yesterday also, and again I am repeating it. It's a very difficult task. Sometimes it takes years together to train a person in maintaining the animals. You need everyday surveillance, you need everyday upkeep of the animal house. Whether you take tea or coffee or not, whether you have taken food or not, you are to visit the animal house and you are to look to the welfare of the animals. If the welfare of the animals is maintained, I can very safely say you are going to get very good results. And if your animals are in bad conditions, you are going to get very bad results. They are not going to be reliable, and the whole experiment or the whole practice or the whole effort fails. So you can go through such type of information. Maybe it is available on net or on website. But the information that I have given you comes out of my own experience working on these animals for last 5 decades. And I have loved my animals, and they have always, they never disappointed me. I always got reliable results, very good results, and those results were duly published also. So the animal is to be respected, animal is to be loved. I wish you all best. If you are going to make some experiments on animals, I think these tips will be helpful to you. Best of luck.